Welcome back to the conference. So we'll start the afternoon session, and it's a great pleasure and honor from my side to introduce Dr. Bruno Sainz. He's an senior investigator at University Autónoma de Madrid in Spain, and is also affiliated with the Department of Cancer Biology and the Institute of Biomedical Investigation in Madrid. He leads a team that is focused on the investigation of the role of cancer stem cell and the interaction with the immune system, in particular the role of immune system to pro-cancer, the role of immune system in promoting the cancer development and the cancer growth, and with a special focus on pancreatic cancer. So today his presentation is titled Exploiting Oxidative Phosphorylation to Promote the Stem and Immunoevasive Properties of Pancreatic Cancer Stem Cell. So please, Bruno.
Thank you very much, Cristina. Okay, you can see the presentation well?
Yes, I can see. Yeah,
okay, yes. Perfect. Well, thank you for the introduction, Cristina. And before I begin, I have to thank Ida, as always, for putting together this Congress, especially this year, a very complicated situation. And she managed to pull it all together and put together the virtual package of the conference. Hopefully in 2 years, we can all see each other again, as we did 2 years ago. And so I'm looking forward to that. So as always, thank you, Ida, for excellent work. So with no further ado, what I'm going to present to you today is a platform and a system that we just recently published in Nature Communications, how we can exploit oxidative phosphorylation to enrich for pancreatic cancer stem cells, and how that gives us ideas also about their immunoevasive capacities. Let me put on the pointer. Okay. So I think it's important to stress why we are all here. We're all here because we know that 8 to 8.2 million individuals die each year from cancer, and this makes up approximately 13% of all worldwide deaths. We also know that 14.1 million new cases of cancer are diagnosed each year. And these are certainly alarming statistics, and experts predict that the situation is not going to get much better. We're expecting a 70% increase in the number of new cases of cancer worldwide in the next 2 decades, which translates to about 21.4 million new patients for 2032. Now, not all numbers are bad. If we look at mortality, in fact, we've seen a 17% drop in mortality over the last decade. So these are actually positive statistics. We've seen this particular in stomach cancer, and breast cancer, and prostate cancer, and colorectal cancer, this tendency to a drop in mortality. Fortunately, we can't say the same for all cancer entities. For example, lung and pancreatic cancer, these are 2 types of cancers where we've seen a continuous rise or no reduction in overall mortality. Now, I think a lot of the positive effects that we've seen in prostate cancer, colorectal cancer is largely due to better screening campaigns and better diagnostics that together have improved overall survival for many different types of tumor entities. But as I mentioned before, the same cannot be said, for example, for lung, liver, and pancreatic cancer in particular, where the 5-year overall survival rates for these cancers are still extremely too low for what we would like to have. In particular for pancreatic cancer, which hovers around 5 to 7% based on the continent or the country. Now, as Cristina mentioned, in my laboratory, we work on pancreatic ductal adenocarcinoma. And in fact, in this entire session, we're going to hear a lot of talks about pancreatic cancer stem cells. So I'm very, very excited about this session in particular. I think we'll learn a lot. I will get the introduction on pancreatic cancer. I'm sure others will follow with more details. As we all know, it's a malignant neoplasm of the exocrine pancreas. It is currently the 4th most frequent cause of cancer-related death, expected to become the 2nd by 2032. It is one of the most deadliest forms of cancer, as I mentioned before, a 5-year survival rate of less than 7%. And I think there are 2 important features about pancreatic cancer that we need to highlight. PDAC, or pancreatic cancer, is usually diagnosed at very late stages, meaning that when a patient is diagnosed, the patient usually has advanced pancreatic cancer with metastasis to secondary organs, including, for example, here the liver. And this is primarily due to a lack of early symptoms. And another feature about pancreatic cancer that we always have to take in consideration is it's a tumor that's highly resistant to chemotherapy and radiotherapy. So it's a challenge to treat pancreatic cancer. In fact, when we talk about treatments for pancreatic cancer, they haven't really changed over the last 20 years. We've had an introduction in the last 10 years with the introduction of, for example, FOLFIRINOX for a select group of patients, and also the combination of Abraxane with gemcitabine to treat pancreatic cancer, which has improved overall survival, not where we would like it to be, but for example, FOLFIRINOX patients can reach up to 1 year overall survival, and gem and Abraxane not much more than 6 months. And I think our failure to properly treat pancreatic cancer for the longest time has been based on the idea that we've thought that all the tumor cells within, all the cancer cells within the tumor were the same. And we know now that that's not the case. We know that not all tumor cells are not the same. There's in fact a hierarchy that exists within the tumor, and there's a heterogeneity that exists at the cancer cell level. And when we think about heterogeneity, and it's something we always need to keep in mind, we always need to be thinking about heterogeneity and how that plays a role in tumor development. We always think obviously about intratumoral heterogeneity, but we can't forget about intratumoral heterogeneity. Now within a tumor, different subpopulations of cells can coexist. We know the cancer stem cell exists, daughter cells exist, other tumor microenvironment cells, and these cells adapt to the tumor environment in and of itself. They adapt to pressures that they're constantly receiving, chemotherapies, for example, lack of nutrients, lack of oxygen, hypoxia, and there's always a constant need for evolution. So this state is very dynamic, it changes, and the heterogeneity can be different today and can be different tomorrow. It's something we always need to keep in mind. And I think if we keep that in mind and we think about therapy, not all cells obviously will respond to the same chemotherapy therapeutic agent because of this heterogeneity. So after treatment is finished, we can usually have a population of cells that survive treatment. And these are the cells that can give rise to disease relapse and the reappearance of the cancer. So I think we all need to start thinking about ways to better treat cancer, and we need to think about different novel ways to combat cancer. We need to, on the one hand, either invest money, time, and resources in developing better drugs, compounds that can be used to inhibit cancer, or at the same time, we need to think about whether or not the cell we are targeting is the cell that we should really be targeting. And this really brings up the idea of, should we be targeting the cancer stem cell as a means of eliminating cancer? And I think Shalmali did a very nice job in one of the earlier sessions discussing anti-cancer stem cell treatments, which I think is a future. It's where we're going. We're not there yet, but soon we should be. Now, in this context and in this forum, I'm not going to talk about the cancer stem cell concept. Everyone here is an expert in cancer stem cells. I just want to reiterate what we now know as the bidirectional cancer stem cell model. Up until about 5 or 6 years ago, we were really considering this a unidirectional model, and based on studies in colorectal cancer with LGR5 positive cells, some very elegant studies published in 2017 in Nature, really shows that this is now a bidirectional model, and plasticity exists across tumor cells. We can consider this model where the cancer stem cell resides in the cancer stem cell niche. It receives cues from the microenvironment to maintain its stemness, in quotes, and the cancer stem cell sits at the apex of the hierarchy. And the cancer stem cell will give rise to the other tumor cells present within the tumor, for example, transient amplifying cells, hybrid cancer cells, and more differentiated tumor cells. Now, what we've learned over the last 5 years is that their plasticity exists. These transient amplifying cells or hybrid cancer cells can dedifferentiate and take the place of the cancer stem cell if the cancer stem cell is targeted and eliminated. So if we have therapy that targets and specifically or uniquely eliminates the cancer stem cell, the transient hybrid cells can receive cues from the microenvironment and now undergo an epigenetic reprogramming, which will induce their dedifferentiation, and they can convert into cancer stem cells, taking the place of the lost cancer stem cell. Now, we can't forget that there might also be quiescent or dormant cancer stem cells within the tumor. Maybe we haven't even identified these cells because we don't know the markers that they express on the cell surface, and they can also respond to these cues and take the place of the cancer stem cell if it's eliminated. So with this in mind, in theory, only combinations of therapies that target the cancer stem cell, plastic hybrid cells, and the tumor microenvironment will ensure tumor eradication. And one of the focuses of my laboratory is to try to discover new anti-cancer stem cell therapies that can be used to eliminate cancer stem cells. Now, I bring to your attention this very nice review that was published in January of 2019, last year, by Sagan et al., where they did a nice recapitulation of anti-cancer stem cell therapies that are in different clinical trial phases. And you can see here, this is just one of the tables of the many tables in the review where they have selected emerging therapies for cancer stem cells. And I think what I can highlight from this table is that the majority of them are still in early phase clinical trials. Many in phase 1, phase 2, and a few in phase 3. So I think we're still far off from really using or introducing cancer stem cell therapy in the clinic, but we should continue our efforts in discovering new anti-cancer stem cell therapies. And there's many ways that we as basic scientists or translational scientists can help in this field or in this approach. Now, there are many pipelines that can be used for discovering anti-cancer stem cell therapies. Many that are used in pharma, but I think a few that we use routinely in the laboratory. And I'm going to discuss two because at the end of this talk, I'll show how our platform can be used for this purpose. One of the more common approaches that we use in the laboratory for anti-cancer stem cell therapy discovery is our enrichment platforms, basically modifying the tissue culture system so that we enrich for cancer stem cells and we can test our anti-cancer stem cell therapies against these cells. And one of the most common ones used by members in the laboratory or many different laboratories are the spheroid cultures, sphere formation assays. We know in adherent cultures, for example, this is an adherent culture of pancreatic ductal adenocarcinoma cells, we have a low percentage of cancer stem cells. If we use CD133 here as the marker, about 3.44%. And if we dissociate these cells and we put them in conditions that will enrich for, that will promote the formation of spheres, so these are commonly known as non-adherent conditions, we remove adherence, we remove also FBS, and these cells over time, over the course of about 5 to 7 days, can form spheres. And in these spheres cultures, what we do see is an increase in the cancer stem cell percentage of cells. Here, we see an increase from 3.44% adherence to 74% in sphere culture. And in these conditions, what we can do is actually perform add anti-cancer stem cell compounds and do endpoint assessments to determine whether or not the anti-cancer stem cell compound has affected the cancer stem cell compartment. Now, these are good assays, but they're not perfect. There are downsides to the 3D cancer stem cell sphere cultures. For example, it's a bit labor intensive. This is a 3D non-adherent system, which it makes it a bit difficult sometimes to, if you need to add drugs, remove drugs, wash the cells. The fact that these cells are in suspension makes it a bit laborious or labor intensive to do these types of manipulations. They do require specific culture medium. There's a short window of time to assess drug efficacy, usually anywhere from 5 to 7 days while the cells are forming spheres, because they do reach critical mass. These spheres will eventually have to be passaged. If not, they will form necrotic cores and will need to be trypsinized and passaged into serial generations. So while it is a good system, it does have its limitations. Other ways to enrich for cancer stem cells is to use the markers that are present on their cell surface. So there are many markers that have been described for different types of cancer. Some that are common amongst different tumor entities are, for example, EpCAM and CD133. We in the laboratory have discovered other cancer stem cell markers for pancreatic cancer stem cells, for example, autofluorescence. This is a marker that we published in 2014 in Nature Methods, where we discovered that pancreatic cancer stem cells and also some gastrointestinal cancer stem cells contain within their cytoplasm discrete vesicles, intracellular vesicles that are coated in ABCG2. And these pumps, what they do is concentrate the autofluorescent vitamin riboflavin inside of these vesicles, which give off an autofluorescence, which can be identified via fluorescence microscopy or by flow cytometry. We've also discovered that cancer stem cells, for example, express CD47 on their cell surface, which is an immunoevasive receptor, P2X7R and FPR2. And we recently discovered that pancreatic cancer stem cells also express anthrax toxin receptor 1 on their cell surface, and we're always looking for new cancer stem cell markers. I think this is important. It's been reiterated in the talks from this morning that it's always important to find new markers. The more markers we know, the better we'll understand the cancer stem cells that are present within the tumor. Now, we've used these markers to enrich for cancer stem cells, and we've tried to perform screening campaigns to test anti-cancer stem cell drugs against the cancer stem cell population via flow cytometry or via FACS sorting. So for this particular experiment, what I'm going to show you is we use CD133 and autofluorescence to isolate the cancer stem cell population from our adherent cultures. And you can see right after sorting, we had a very good high percentage of CD133 positive cells and autofluorescent cells. The problem is when you put these cells back into adherent culture, they quickly dedifferentiate. They quickly differentiate again into non-cancer stem cells. We lose the ability to identify these cells based on the expression of these markers. And basically the culture after about 3 or 4 days recapitulates the heterogeneity or the distribution that was present prior to sorting. So this is a limitation. You have a very short window of time where you can actually test anti-cancer stem cell therapies on sorted cells. So we asked, is there any way that we can establish, in particular in pancreatic cancer, a 2D adherent system where we can maintain an enrichment in cancer stem cells? And we went back to our previously published RNA-seq data. And this was RNA-seq data performed on cancer stem cells isolated from 6 different PDX models. So these are different tumors. Arbitrary nomenclature. We established primary cell lines from these PDX models, and we can isolate the cancer stem cells. And what we discover are thousands of genes that go up and thousands of genes that go down in the cancer stem cell compared to the non-cancer stem cells. And we looked at pathways that were enriched in cancer stem cells. And we published a few of these pathways. One that I want to stress today is a publication in 2015 by Sainz et al., where we showed that the cancer stem cells versus the non-cancer stem cells overexpressed genes that were associated with metabolic processes. You can see here on the right, the cancer stem cells, on the left, the non-cancer stem cells in this heat map. And the cancer stem cells upregulate genes involved in lipid metabolism, TCA or the Krebs cycle, mitochondrial oxphos components, indicating that these cells could be depending more on mitochondrial respiration than traditional glycolysis, which is what we saw in the non-cancer stem cells that upregulate genes involved in glycolysis, as you would expect. Now, in this paper by Sainz et al., it was shown that indeed pancreatic cancer stem cells are oxphos dependent, and this is the mechanism they use to obtain to meet their energy requirements. So indeed, we can differentiate cancer stem cells from non-cancer stem cells based on the expression of markers, based on the cells that can form spheres and cells that cannot form spheres, other biological properties. We've really focused on how cancer stem cells differ from non-cancer stem cells at their metabolic level. So we've shown, or our hypothesis or the basis of our studies are based on the fact that cancer stem cells have functional mitochondria or more functional mitochondria, and they use oxphos to meet their energy requirements, while non-cancer stem cells could have dysfunctional mitochondria and rely more on glycolysis to meet their energy requirements. I think a picture's worth 1,000 words, and you can see here, this is electron microscopy performed on cancer stem cells isolated from PDAC cultures. We used here the marker CD133 to isolate cancer stem cells, and these would be the CD133 negative cells, which would be the non-cancer stem cells. And you can see here, and the cancer stem cells are very textbook mitochondria, well-defined cristae, which would indicate functional mitochondria, while in the non-cancer stem cells, we have very swollen mitochondria, not very well-defined cristae, which could indicate dysfunctional mitochondria. You can also see here when we isolate cancer stem cells here in this particular experiment using CD133 and autofluorescence as our markers to separate the cancer stem cells from the non-cancer stem cells. And we had different probes, for example, deep red, which is a probe for mitochondrial mass, CMX-SWAS for mitochondrial membrane potential. And you can see here that the cancer stem cells have much higher staining for these probes than the non-cancer stem cells. Here we've shown the quantification of the staining in 2 different PDX-derived cell models, PANC1A5 and PANC-A6L. Also, if we look at mitochondrial DNA copies, we see that the cancer stem cells have more mitochondrial DNA. So no, they don't just have more mitochondrial mass, but also more mitochondrial DNA, again, indicating more mitochondrial biogenesis and most likely more efficient functional mitochondria. So we thought, can we exploit this oxphos dependency that cancer stem cells have to create a system where we can have that are enriched in cancer stem cells? Now, the non-cancer stem cells, when glucose is shuttled into the cell, it's converted to glucose 6-phosphate and to pyruvate and converted to lactate, shuttled out of the cell. And this gives rise to ATP molecules, but also gives rise to a lot of building blocks for proliferation. So cells that are highly proliferative, that need energy fast, that need energy and building blocks quickly, glycolysis is really the way to go for them. They will survive. They will quickly obtain their ATP and building blocks that they need because of their highly proliferative state. Now you could say the cancer stem cells, they don't mind taking their time. They don't need energy that quickly. They don't need these building blocks because they're not very highly proliferative cells. You even could call cancer stem cells quiescent slow cycling. So glucose 6-phosphate is converted to pyruvate and then shuttled into the mitochondria, into the TCA cycle, and then the electron transfer chain can give rise to 36 ATP molecules to meet the energy requirements of cancer stem cells. So we really thought, how can we get rid of these cells and maintain these cells using OXPHOS? And we did something extremely simple. All we did was change the sugar source or the carbon source from glucose to galactose. So galactose also enters into the cell. It's also converted into glucose 6-phosphate, but via several more limiting enzymatic steps. Okay, so this process it takes much longer. In fact, Warburg in his day actually showed that cells, when given galactose, use glycolysis 18 times less when they're given galactose compared to glucose. So this process is, you could say, a much longer process. Cells that need energy and building blocks very quickly will die in the presence of galactose. And what you'll do is enrich for cells that have functional mitochondria, and then you can shuttle pyruvate into the mitochondria and use OXPHOS for their energy requirements. So this was our approach, and this is what we published just a few months ago in Nature Communications. Want to highlight here, this is all the work was from Sandra Valle, who was a PhD student in our laboratory. So this is the basis of her PhD thesis, and which is the platform I'm going to show you in the next 10 minutes or so. So one of the first things we did was to culture our cells in glucose or in galactose. And we culture our cells in 5 millimolar glucose or galactose because these are physiological relevant sugar levels. Media was changed every 1 or 2 days. And what we saw very early on in the presence of galactose is we saw a lot of cell death, a lot of apoptosis. Very early on, we determined that these are the cells that do not have functional mitochondria that were dying in the presence of galactose. And then we eventually established a very manageable cell culture. The proliferation rate of this culture was very different than this culture, as you can see in this representative image, but these cells could be trypsinized, these cells can be frozen, they can be thawed, they can be passaged. It's a viable cell culture system. At the level of proliferation, obviously there's a difference. We measured proliferation in real time using the in situ site Zoom system, and you can see when these cultures were placed inside the system over the course of 40 hours, the cells, the glucose cultured cells proliferated as would be expected, while the galactose cultured cells essentially had no proliferation over the course of 40 hours. The system also has fluorescence, so we were able to look at the autofluorescent cells to look at these vesicles present within the cell culture. And we could determine a ratio between autofluorescent cells and confluence. And basically what we determined is that these cells that were present in galactose were essentially all autofluorescent positive cells. So we had a significant increase in the autofluorescent positive cells, our first indication that we could be enriching for cancer stem cells. Now, I think it's important to point out that we've gone as far as 70 days maintaining these cultures in galactose. We haven't gone out any further there was no reason to. And in fact, we can establish these cultures from freshly digested PDX, PDXs, which could highlight its translatability to perhaps establishing these types of cultures directly from a freshly digested patient tumor. Now, we obviously needed to show first that the system is working as we expected. Are these cells do these cells have increased OXPHOS? One of the first things we did was to look at mitochondrial activity. So we used different probes to look at mitochondrial mass. Here, this would be MitoTracker Deep Red and MitoTracker MitoBlue to look at mass, probes to look at membrane potential, and probes to look at ROS production. And in all of the cases, the galactose cultured cells had an increase in mass, membrane potential, and ROS compared to the glucose cultured cells, as we would expect. And using these probes also via immunofluorescence, I think it's clear. You can see very, very nicely that the galactose cultured cells have increased mitochondrial mass, have increased membrane potential compared to the glucose cultured cells. Now, to really test if these cells are OXPHOS capable, it's important to measure the oxygen consumption rate. And so for this, we use the Seahorse system, the MinoStress test. I won't go into the details because Daniel Gephardt at the end of this session will probably give us a very nice presentation on this technology. But we use the Seahorse system to measure both oxygen consumption rate and extracellular acidification rate as a measurement of glycolytic potential. And you can see here in the blue bars, we have the galactose cultured cells, in the red, the glucose cultured cells. And you can appreciate here more oxygen consumption in the presence of galactose versus glucose. And the two parameters which are quite important are basal respiration and maximal respiration. And you can see here in both cases, the galactose cultured cells have more basal and maximal respiration compared to the glucose cultured cells. We saw the opposite with ECGAR, as would be expected. We have a decrease in extracellular acidification rate, blue compared here to red, and which would indicate less glycolysis, and which we measured in a separate analysis by measuring lactate production, and the galactose cultured cells produce less lactate than the glucose cultured cells, as would be expected, since they're more OXPHOS dependent. So the system in theory works. It's working in the way we expect it to work, simply by adding galactose in lieu of glucose as a carbon source. We can enrich for cells that are OXPHOS dependent or use OXPHOS in a 2D system. Now the question is, are these cells enriched in cancer stem cells? And these are the next few experiments I'll show you, where Sandra did a lot of experiments to validate this. I'll just show you a select sample of experiments that were performed. One of the first things she did was obviously to look at the expression of cancer stem cell markers. She looked at a battery of different cancer stem cell markers. Here are just some representative flow cytometry plots looking at autofluorescence, anthrax toxin receptor 1, and CD133. And what you can appreciate in the quantification, this is several samples across 4 PDX models cultured in the presence of galactose or in the presence of glucose. And in all the cases, we saw an increase in the cancer stem cell markers in the galactose cultured system versus the glucose culture system, sometimes 7-8-fold increase in these cancer stem cell markers. We also looked at self-renewal employing the very typical sphere formation assay to measure self-renewal over 2 generations, first and second generation, and in all cases saw that spheres cultured in the presence of galactose form more spheres than spheres cultured in the presence of glucose. And if we take these spheres and we digest them, and we look for the presence of cancer stem cell markers in these spheres, we see an increase always in first and second generation, in autofluorescence, or also in the double positive cells CD133 and CXCR4, which have been shown to, in fact, isolate or be able to identify the metastatic cancer stem cells in pancreatic cancer. At the level of gene expression, and we already heard our previous talk on NOC4, part of this battery of pluripotency-associated genes, we saw increases always in the galactose cultured cells versus the glucose cultured cells. My laboratory has always been interested in NANOG, and in collaboration with Patrick Herman at the University of Ulm, we performed experiments where the cells were infected with a lentiviral vector that has the NANOG promoter driving yellow nanolantern, YNL, it's a fluorescent protein. And we could see that if we placed these cells in glucose and galactose, the percentage of YNL positive cells increased in galactose, the presence of galactose versus the presence of glucose, again, indicating an enrichment in cancer stem cells in these cultures. We also performed RNA-seq analysis, and of course, as you would expect, we had hundreds of genes that went up and hundreds of genes went down. We performed gene state enrichment analysis to find pathways associated to these modulations in gene expression. And we could see in the galactose cultured cells, enrichment in pathways, for example, mTOR and IL-6 JAK-STAT signaling, which have been already shown to be important for cancer stem cells and pancreatic cancer. And as you would expect, we have an enrichment in genes involved in oxidative phosphorylation. We also saw pathways that were downregulated in the galactose culture cells. For example, G2M checkpoint, E2F targets, which could tie into this more quiescent, slow proliferative state that we see in our cells. And also interestingly, we saw a downregulation in MYC target genes. So the MYC pathway was downregulated in the galactose culture cells. We've shown previously that MYC is downregulated in pancreatic cancer cells. And I think in this system, it's important because we know that MYC is a negative regulator of OXPHOS. It actually promotes glucose glycolysis over OXPHOS. So this makes sense in our setting. We also performed tumorigenicity assays using extreme limiting dilution assays. And you can see in all cases that at all dilutions, the galactose cultured cells outperformed the glucose cultured cells, giving rise to a higher cancer stem cell frequency in these cells, in mice injected with these cells. Fortunately, what we used were H2B M-cherry labeled cells. So we're able to digest these tumors and look within the M-cherry positive population to see whether or not we also have an increase in CD133 positive cells. And we saw across almost all of the tumors digested that there was an increase in CD133 positive cells in tumors that arose from cells that have been treated with galactose versus cells that have been treated with glucose. Again, indicating an enrichment in the cancer stem cell population. We also examined metastasis, the capacity of these cells to metastasize in two different in vivo models. We used the zebrafish model and also an immunocompromised mouse model. In the zebrafish model, cells were injected, cells that were treated either with cultured either with glucose or galactose were injected intravenous injection into zebrafish. And the fact that they were labeled, we could see their migration, proliferation, and expansion in the tail of these zebrafish. And you can see over the course of 6 days that the galactose cultured cells had a higher capacity for proliferation and forming these microtumors compared to the glucose cultured cells. And this is just a quantification of this data. In the mouse system, what we use are immunocompromised mice. So these mice have no adaptive immune system, but do have an intact innate immune system. And we did intravenous injection of these cells. And we examined these mice at short times, so 10 days post-injection and 3 months post-injection. And at 10 days post-injection, what we saw, we didn't really see a difference in the capacity of these M-cherry positive cells to disseminate and reach different organs like the spleen, liver, pancreas, lungs. We did see more cells in circulation. There were more M-cherry positive cells in blood from the galactose cultured cells, and also an increase in cells present within the pancreas. What we really did see a difference was later on, 3 months post-injection. What we saw is a higher capacity of these cells to seed and remain in the spleen, liver, pancreas, and lungs if they were previously cultured with galactose versus glucose. You can see here in this BLI analysis, essentially all of the tissues that we analyzed were positive for M-cherry. Indeed, these cells prior to injection do upregulate genes that are involved in EMT. So they do have higher expression of slug, snail, Vimentin, ZEB1, which could answer why these cells had a high capacity to disseminate and to form micrometastases, for example, even in the liver of mice. So here we have cytokeratin 19 staining of livers from mice injected with glucose culture cell or galactose culture cell, and we're able to detect far more micrometastases in the livers from these mice versus livers from mice injected with glucose cultured cells. So this was a very high capacity for dissemination, which really caught our interest, that they were so efficient at disseminating and surviving for such a long period of time. And there have been studies over the last 5-10 years linking metabolism to immunoevasion and how metabolism can actually regulate the expression of immunoevasive markers. So we wanted to see whether or not our cells also had this immunoevasive capacity. So we looked at a number of markers, cell surface markers that have been linked to immune evasion. PD-L1, CD47, which is a don't eat me signal for macrophages, and also proteins that have been linked to interactions with NK cells, ULB2, 5, and 6. And this is the quantification of all of our cytometries across 4 different PDX models. And you can see in all cases, we have an increase in PD-L1, CD47, other markers that could be considered important for immune evasion, CD155 and CD206, and a downregulation of these proteins or ligands that can interact with natural killer cells. So downregulation here and upregulation and the other markers, indicating perhaps a true immunoevasive phenotype. Now, we can test this ex vivo, in vitro. So one of the first things we did is to test whether or not macrophages could phagocytose glucose culture cells versus galactose culture cells. And this is based on a study that we published in 2015 in Clinical Cancer Research, showing that cancer stem cells overexpress CD47. And this is a marker, this is a don't eat me signal. So the more expression of CD47, the less capacity macrophages have to phagocytose these cells. So the experiment that we set up was stain our cancer cells cultured in the presence of glucose or galactose with pKh67, which basically gives it a green fluorescence, and co-culture these cells with macrophages stained with DILC, which basically we have red macrophages, we have green tumor cells. And these cells were previously cultured in the presence of glucose or in the presence of galactose. And then we co-culture these cells together, and the idea is that if the macrophages phagocytose the green cells, we will have a double positive population that can be determined or quantified by flow cytometry. We see here the double positive population. So these are the macrophages that have phagocytosed the glucose cultured cells. Now, what do we see with the galactose cultured cells? A decrease, about a 50% drop in the capacity that these macrophages have to phagocytose these cells. We validated this in macrophages established from 3 different donors, and we used 3 different PDX models cultured in the presence of galactose or glucose, and in all cases, the macrophages were less capable of phagocytosing the galactose cultured cells versus the glucose cultured cells, indicating that you have this immunoevasive phenotype at the level of macrophage phagocytosis. Also, since we saw overexpression of PD-L1, we wanted to see whether or not activated T cells were less or more capable of inducing cytotoxic cell death. Our galactose culture cells had an overexpression of PD-L1. So what we did is culture these cells and also the glucose culture cells with activated T cells, and we measured cytotoxic induced cell death. And we saw across T cells from 3 different donors, the capacity that T cells had, these T cells were more efficient at inducing cell death of glucose cultured cells than galactose culture cells. Again, suggesting that our galactose culture cells are indeed upregulating receptors proteins that are helping them evade the immune system. So I think this is really the first time where an extensive study of the immunoevasive properties in cancer stem cells and pancreatic cancer has been performed. And I think it's also interesting because it links very well that the OXPHOS metabolic capacity of cells can be can potentially be linked to immunoevasive properties. So I think there's a lot to be learned from this. There's still a lot more to be done, obviously, but this is something we're exploring in the laboratory because we find this extremely fascinating, and it could provide more insight into the development of future immunotherapeutic drugs, perhaps using immunotherapy in combination with metabolic inhibitors. So with this, I would like to end and conclude the conclusions that we've generated from this study. I hope that I've convinced you that PDX cells can be efficiently cultured in galactose. In lieu of glucose, these cells survive, they can be passaged and cultured, and they are less proliferative than glucose culture cells, but it is a feasible cell culture system. These cultures enrich for cells with higher mitochondrial mass, membrane potential, oxygen consumption. These cells are using OXPHOS to meet their energy requirements. Galactose culture cells are enriched in cells that express traditional cancer stem cell markers. They're also enriched in cells that have higher self-renewal potential and increased expression of genes linked to cancer stem cell pathways. Also, they have downregulation of genes that are also involved in pathways like MYC, which are downregulated in the galactose cultured cells, which I think is also very interesting and fits with our previously published studies. Galactose culture cells have higher tumorigenic and metastatic capacity in vivo. We've shown this in several in vivo models, and galactose culture cells upregulate cell surface receptors and proteins consistent with an immune evasive phenotype. So we are using this platform in the laboratory to study the biology of cancer stem cells, et cetera, but we also want to use this platform in the laboratory at the level of cancer stem cell drug discovery. And I think this platform is very amenable to cancer stem cell discovery. You can envision taking your 2D adherent culture, trypsinizing the cells and seeding them in the presence of galactose or in the presence of glucose. Here you'll achieve a cell culture that is enriched in cancer stem cells, and here you will have a cell culture that is more heterogeneous, low percentage of cancer stem cells, but also representation of the other cells, the hybrid and the terminally differentiated cancer cells. And here you can perform your anti-cancer stem cell compound screening analyses or campaigns. I think this system has an upside to the other systems that I explained earlier in the talk, that it's extremely low maintenance. It's a 2D adherent nature, so it's very easy to add drugs, remove drugs, wash these cells, but manipulate these cells without worrying about the cells being removed or washed away because of the non-adherent state that other systems have. There's no complicated culture medium required. Basically, just switch out glucose for galactose. You have a long window of time to assay drug efficacy. It doesn't have to be done within a short period of time. Once these cultures are established, they can stay in culture for 60, 70 days. It allows for the simultaneous identification of inhibitors that are targeting cancer stem cells and non-cancer stem cells. Perhaps you want an inhibitor that targets both populations, or perhaps you want one that is specific. This system will allow you to discriminate between those two. And of course, the system can be adapted to co-culture experiments. We can introduce into the system CAFs. We can introduce into the system immune cells, and we could potentially discover new immunotherapies, for example, with this platform. So with that, I would like to end. Of course, I have to acknowledge the members of my laboratory at the Biomedical Research Institute in Madrid, especially Sandra Valle, which is, as I mentioned earlier, this is the basis of her entire PhD thesis project that she defended earlier this year. Also, my collaborator, Miguel Fernandez Moreno at the university, who is co-corresponding author on this study. We have several collaborators. None of this work can be done without collaborators, in particular Patricia Sancho, who is at the University Hospital Miguel Servet, who will also be giving a talk, the first talk after the coffee break. So I'm very excited to hear her talk as well, but she's been an essential collaborator in our studies. As I mentioned earlier, we have a collaboration with Patrick Herman at the University of Ulm, with Kanishka and Caroline in his laboratory, who have helped us with the NANOC promoter studies. Our medical collaborators, and of course our collaborators who have helped us with all the zebrafish experiments. And of course, we always have to end thanking our funding sources, because without them, we could not perform these studies and these experiments. And I thank you all for your time, the organizing committee for selecting our abstract for a talk, and for your attention. I'll be more than happy to answer any questions. Thank you.
Okay, thanks a lot, Bruno, for this very elegant and very nice presentation. I will start with a couple of questions I see here in the chat. So we have a question from Farah Hussein, who's saying, thank you for the very nice presentation. How do you predict the GAL-induced cancer stem cells enrichment for other solid tumors?
Good question. And this is something we would definitely like to do. We haven't gotten around to doing it yet. We focused on doing the entire first system in PDAC with many different PDX models. I would suggest that I would think it would work. I would especially think it would work for tumors that have been shown. There are other tumor entities where it's been nicely shown that the cancer stem cells are also OXPHOS dependent. So I would expect this will work. This will work the same.
Okay, very good. And we have another question from Abiola Abdularalam Ainalaja. What is the composition of the galactose culture medium? Any increase in glutamine, sodium pyruvate, or sodium bicarbonate?
Sodium pyruvate, glucose, as I mentioned, 5 millimolar. So I think that's 0.9 grams per liter, if I'm not mistaken. And then everything else you would normally add to your culture medium. We use RPMI, 10% FBS, antibiotics, glutamine, but basically all you do is you have to buy the medium without, it's DMEM without glucose, and then adding your galactose. Everything else is the same. It's a very easy system.
Okay.
Adi?
Okay, we have
There's another question.
From Adit, she was raising her hand.
Yes. Okay.
Yeah, okay. I wanted to ask you two questions. One of them, if I'm not mistaken, I've noticed that in the galactose grown cells, the spheres were smaller than, so what is the explanation for this? If they're supposed to be cancer stem cells?
Exactly, they're supposed to be cancer stem cells, of course. Now, the bigger the sphere doesn't necessarily mean the more cancer stem cells. The more spheres would indicate that the system from the very beginning enriched for cancer stem cells. We believe that the galactose system, which is what we saw at the level of, so I've been presented all the cell cycle analysis that we've done, and we also did analysis to look at to look at PKH, the maintenance of PKH 67 staining in the cell membrane. All of this indicates that these cells in the presence of galactose are more slow cycling, less proliferative. I don't want to say quiescent, but they definitely have, their proliferation is much lower. So I think the number of spheres represent the number of cancer stem cells that are there, but I think the size of the spheres might have to do with their proliferation rate.
Okay, and the second question is about reversibility. If you take the cells that were
Yes.
grown with galactose and put them back in glucose,
they take off. Complete plasticity. The cancer stem cell markers go down, the proliferation rate goes up. Everything that you would expect with traditional plasticity occurs. Very good question.
Thanks.
Okay. So we have another question here in the chat. Okay, there is a comment from Claudia Matucci. Great talk, Bruno. This model can give you insight for cancer stem cell research for other tumor models. And I guess you confirm that you are going to investigate.
Yes.
Also
but
another.
Feel free, anyone to investigate.
And then, okay, we have another question from Aisel Kuzera, who is asking thank you for the great talk and tremendous work. How would you explain upregulation of mTORC1 but decrease in proliferation when they are fed with the galactose?
Yes, there's still some things we cannot reconcile. Now, I think the upregulation of mTOR fits with what we know about mTOR and mitochondrial biogenesis and metabolism, so that makes sense. How it could go against the downregulation or the cell cycle data we have. I think we have yet to completely explain that. Again, what we have to keep in mind is that these gene enrichment analyses basically take, these gene ontologists can have hundreds of genes that are related to a pathway, and if you reach significance, meaning that if enough genes are significantly expressed out of the list of genes that are related to that pathway, you can say you have an activation of that pathway. But we really have to look at all of the genes to really see which are the ones that are playing a role. We can just say that pathways enriched, but we haven't gone and looked at the specific genes.
Right.
And I think if you want, I can also answer some of these questions outside of the time if we're going over on time.
I mean, there are still a couple of questions, but for the sake of time, maybe you can answer in the chat, and others can still submit the question. So thanks a lot for the great presentation. Congratulations.